Okay, our next podcast is going to look at a couple of different things, some things related to genetic engineering and the expression of certain genes. So in this, we'll start by talking about gene expression. So genes can be expressed, and that means they're either activated or non-activated. There's different things that can kind of control this. Um, and One example we can look at is the idea of cloning and taking out the genes, all of the genes from one cell, and then implanting them in another cell. And So this shows the technique of cloning that is done. This was first done in 1997, I believe, and they cloned a mammal, the first mammal that is that was cloned in 97. Um, uh, the famous Dolly the sheep. So Dolly was the first um, mammal that was cloned. Now it actually died prematurely, but this is the technique that was used, and it's still used in cloning processes today. So we take cultured mammary cells from a donor, and we take an egg donor cell. So we take egg cells from this individual right here. The egg cell, the nucleus is extracted, so it's just a blank egg cell, and then we culture these two cells together, and we get the nucleus from this mammary cell will go into the egg cell, and now we have one egg cell with a new complete nucleus, diploid nucleus. And so it then acts as a zygote and starts to develop and grow. It will divide into two cells, two into four, four into eight, and so forth until it develops into an early embryo. Now, at this point, it has to be injected into a surrogate mother that will go through full embryonic development, and this will produce a clone of this individual because its genetic information is in here. We pulled all the genetic information out of the egg cell, and so this individual will be a clone, an exact physical copy of this individual. And so that's choosing cells to turn on and off. Now, this is also done therapeutically, where um, certain things are cloned for the production of something that is used medically. Stem cells are cells that have not differentiated specifically yet into their particular cell type, and so they can be kind of tricked into being produced into a certain cell type that can be used therapeutically or medically. So here in the example, you can get stem cells either from your marrow or from an early blastocyst, and these cells can be put into cultures. Under certain conditions, they will develop into certain types of cells, blood cells, nerve cells, or liver cells, or whatever, and then these can be used therapeutically. So that's an example of kind of tricking the cell or turning on certain genes based on the conditions that are there. That production, when it starts to develop from a stem cell into carrying out its particular characteristics, that's called differentiation. When the cell takes on its specific structural characteristics and becomes specialized. Now, this is getting certain genes to turn on and off, and the way they do that, the way certain genes turn on and off is through a process called differentiation, through a process called gene expression. And in this gene expression, there is a cluster. There's a cluster of functionally related genes on our DNA, and these are called an operon. The operon is a stretch of the DNA that includes particular sections that can turn on or off other parts of the DNA. So operons are found in eukaryotic cells and in bacterial cells. The bacterial gene, it has one single strand, long strand of DNA. It has these operons on it, as well as eukaryotic cells. So here we see in this this image here, we see our DNA strand all the way down, and there are certain segments that are considered different genes. We have certain genes, when they are turned on, they will produce certain things like a protein here, and this protein may go here and either stop or start or activate other parts of the DNA. So it's like these genes can turn on these other parts of the DNA to get other genes to code. Now in the operon we have these, here's an operon, the trypoperon, it produces tryptophan. So in this operon we have structural genes. These right here are just referred to as the genes of operon. And so these are going to make the polypeptide that's going to be your tryptophan, your protein. And we have an operator. The operator switches transcription on and off. And then we have a promoter right here. This promoter is where the RNA polymerase binds and starts that process that we just talked about of RNA transcription. All right, so those sections are called an operon of the gene. Also, we're going to talk about some techniques that are used in getting this genetic information in kind of practical applications of using some of these genes. So there's a lot of techniques that are out there in what's called genetic engineering, which is just manipulating the genetics to get certain either genes or proteins produced that can be of use. One of the most common ways is a process called recombinant DNA. Recombinant DNA is a technique that kind of identifies and manipulates or isolates a gene that can be manipulated. Usually in this process, plastids are used. Plastids are these round DNA sections that are found in bacteria. So in this case, we take the plasmid out of the bacterial cell, and we can use these. They're easy to manipulate. So in this case, we take the plasmid out of the bacteria. We have a gene of interest over here that maybe a gene that's on the DNA of this hummingbird right here, and that's our gene of interest. Okay, so we can isolate, we can cut and cut this out. We can use different enzymes or ligases to separate that gene of interest and get that out of there. Basically, we cut out a gene from the DNA of this hummingbird and we implant it on the plasmid. So this can be right here. So we have this plasmid. It is cut apart. We have this site right here that's separated, and our new hummingbird DNA fragments are added to it. And so now these are our recombinant plasmids, and those are placed there, and then they can be implanted back into the bacterial cell, and then we can grow those bacteria in culture until we get a bunch of those. And so this technique is used a lot for different things. It's used to make a copy of a gene that is of interest for, agricultural practices use this quite a bit, and human um, therapies use this. It may be to make proteins. Maybe this gene makes a protein that is useful, say, in making a protein that's used by diabetics, insulin or whatever. And so maybe it's not the gene that is going to be collected, but it's the protein made by the gene that is collected. Blood clotting factors that can be made in this fashion. So there's a lot of different applications for this. But this is called recombinant DNA. So recombining the DNA. There's other ways of kind of introducing genetics into a new to get the gene out of a gene of interest out of a cell, and then use it. Electroporation, which involves exposing that DNA to high voltage, can separate it out. Cell fusion, sometimes fusing cells together. Viruses are used. The virus actually extracts genetic information and then can carry it and pull it out, and it can be done that way. Bacterial plasmids is the most common way of this technique, but these other ways are used as well. Let's see. So enzymes are used to either cut these apart, as in right up in here, and then also to bind them back. Different types of enzymes, ligases and enzymes called endonucleases, are used for breaking and hooking these back together. And that's really the main things about genetic engineering that we're going to talk about. There are ethical questions that come up with a lot of the cloning and gene therapies that are involved. And also the Human Genome Project was completed, and this project uncovered all of our sequence of the human genome. And so our understanding of genetics, especially human genetics, has come a long way in the past decade or so. And so some of these techniques are relatively new and newer technologies, but they hold a lot of potential. But there are ethical issues and possible questions, such as genetically modified crops versus natural wild crops and things like that. So that's where we end for genetics and genetic engineering, and that ends our podcasts for this unit.